everyone. I am Rebecca McGehee-Themis, and this is my 3-minute thesis, establishing Glary melanoma as an invertebrate model for Staphylococcus hemolyticus infection and immune response. Let me introduce you to the model. This is Glary melanoma, also known as the waxed worm. He has an interesting attribute that when he is infected with something, the larva produces melanin as part of an immune response, which causes a color change. This allows to be almost an indicator to understand how severe an infection is just based on the color. So what we did is we took our Staphylococcus hemolyticus strain, which is represented as those little purple circles, and we inserted it into our larva, which caused an immune response. These immune cells fought the infection, causing phenyloxidase, which is an enzyme that causes the melanization to occur. And this cycle continues on of them fighting and melanization occurring over and over again until one of two things happens. Either the larva succumbs to the infection, or the larva gets to live another day. In our study, we used a high enough dose that every single one of the larvae did succumb to the infection so that we can better understand it. So that is shown in the survival of waxworm larva graph. Below that is our bacterial burden. What we wanted to know next is if the bacteria was actually reproducing inside of them. So at those, we did the exact same experiment, except at 3 different time points, 4, 8, and 12, we extracted hemolymph, which is essentially the larva's blood. We plated that hemolymph and we let the bacteria grow. And we were able to see that, yes, indeed, at different time points, there was a larger quantity of bacteria, which shows that the bacteria is reproducing inside the larva. What we wanted to know next is what genes, what immune cells are at play, what is actually happening. So we've implemented real-time qPCR, which I currently do not have data for, but basically we will be extracting a tissue sample from the larva at different time points and using our lovely little PCR machine to determine what immune genes or what genes that are associated with different immune responses and immune cells are at play at those different times so we can understand who's actually fighting off the infection. Great. We know the story now. But why is this important? Why are you talking about bugs? Is probably what you're thinking. Well, 6 short years ago, we all lived through a global pandemic, and we came to understand how important the immune system is. And immunology as a science plays a big role in our everyday lives. So we should try to learn as much about it as possible. These guys are invertebrates, which means they only have an innate immune system. The innate immune system doesn't change, it doesn't learn. What's in your DNA is what you get. We have an innate immune system, but we also have an adaptive immune system. And what's valuable about these larvae is that we can study the innate immune system without the interference of the adaptive immune system that's able to change and grow and learn. It could learn this bacteria and fight it off better in the future. But these guys, that doesn't happen, which was what makes them so interesting. Thank you very much. You have a lovely day.